Honorable Vice Principal, respected teachers and doctors, intern doctors, good morning. Welcome you all to the weekly scientific seminar. Today, the seminar is organized by Department of Microbiology and the topic is Antibiotic Resistant Pattern at CBMCB from 2021 to 2023. The seminar will be chaired by Professor Zohra Begum, Professor of Microbiology, CBMCB, and the speaker is Dr. Mahmudur Rahman, Assistant Professor, Department of Microbiology, CBMCB. Now, I request our chairperson, ma'am, to conduct rest of the session, please.
Ramadan Mubarak and good morning. Honorable Vice Principal Sir, respected teachers, learned audience, Assalamu alaikum. I feel great pleasure to have the opportunity to chair the today's session and for this I express my gratitude to Professor Murshid Alam Sir, Head of the Department of Microbiology Department. Ladies and gentlemen, our today's topic, antimicrobial resistant pattern at CBMCB 2021 to 2023. Antimicrobial resistance has emerged as one of the greatest threats to the global public health in the 21st century. The common drugs were used to treat the infection caused by the bacteria, parasite, virus, and fungi have developed resistance, and threaten the effectiveness of the prevention and treatment. So, infections are becoming difficult to treat and resulting in increase in the morbidity and mortality. Today's our speaker is Dr. Mahmoud Rahman, Assistant Professor of Microbiology Department. Now I request our speaker to deliver his presentation before the audience.
Thank you, madam. Honorable Chairman of this scientific session, Honorable Vice Principal Sir, Honorable Professors, and learned audience, Assalamu alaikum. Myself, I am now proceeding this scientific session. Title of the study is Antibiotic Resistance Pattern at CBMCB from 2021 to 2023. Introduction. Infectious disease remain a major health problem in low and middle socio-economic countries, including Bangladesh. From the 19th century, the development of effective antimicrobial drugs has reduced the incidence of life-threatening infection. The modern antibiotic era, with the name of Paul Ehrlich and his idea of magic bullet that selectively target only diseases, causing microbes and not to the host. The first antibiotic penicillin, discovered in 1896, rediscovered by Fleming in 1928, and mass production of penicillin began in 1943. In 1945, immediately after winning the Nobel Prize, warned about the possibility of emergence of resistance as a consequence of misuse of miracle drug penicillin. Antimicrobial resistance is a natural consequence of exposure to antimicrobial drug caused by the inappropriate and irrational overuse of antimicrobials in health and veterinary sector. Irrational use of antibiotic is the greatest driver of resistance. 50% of antibiotics are prescribed inappropriately. 30% of patients have poor compliance. 20% of population do not have access to essential antibiotics. Now, environmental causes of antibiotic resistance by who? They says overprescribing of antibiotics, patient not finishing their treatment, overuse of antibiotics in livestock and fish farming, poor infection control in hospital and clinics, lack of hygiene and poor sanitation, lack of new antibiotics being developed. Who showed the picture? These are the causes of resistance. Now, origin of drug resistance. Genetically, chromosomal mediated resistance is due to mutation in the gene that codes for either target of drug or by transport system in the membrane that controls the uptake of drug. Extrachromosomal mediated, that is plasmid. Plasmid, which contain R factor that carry genes for a variety of enzymes that can degrade antibiotics and modify membrane transport system, and this plasmid allows resistant gene to move between different type of bacteria. Gene transfer occur by following mechanism, that is transposon, transformation, transduction and conjugation. Origin of drug resistance non-genetically, which is contain metabolic inactivity, loss of target structures, that is transposon. National Antimicrobial Surveillance in Bangladesh by IEDCR explain how antibiotic resistance proceed in this picture. Antimicrobial resistance in 2016, who endorsed Antibiotic Awareness Week, took place from 14 to 20 November, acknowledging the global importance of this growing public health issue. All countries are required to develop their own national action plan and start implementing it at local level. Major objective of the action plan is to strengthen the knowledge and evidence base through surveillance and researches, and this is particularly relevant to countries where there is a lack of research. Observation of antimicrobial resistance in Southeast Asian countries. In case of Staphylococcus aureus, more than 50% isolates in hospital are methicillin resistant. In case of Pseudomonas klebsiella and serratia, multidrug resistance persists in overall and causing huge mortality and morbidity. In case of typhoid, MDR Salmonella typhi prevalent all over the region, causes 10% of fatality rate in children. In acute respiratory infection, pneumonia, 69% of Streptococcus pneumoniae resistance to penicillin in Thailand. Starts of antibiotic resistance. History of antimicrobial resistance. Penicillin, introduced in 1943 and resistance in 1946. Streptomycin, resistance in 1959. Tetracycline, resistance in 1953. Erythromycin, resistance in 1988. Vancomycin, resistance in 1988. Methicillin, resistance in 1961. Ampicillin, resistance in 1973. Cephalosporin, resistance in 1964. Now, objective of the study: isolation of common microbial with some of their species at CBMCBs. To identify the antimicrobial resistance pattern of commonly isolated bacteria at CBMCBs during 2021 to 2023 for the last 3 years. To determine the rate of resistance of antibiotic which are commonly used by the community peoples. Materials and method. We use traditional culture method. Type of the study is retrospective observational study. Study period, that is from January 2021 to December 2023. Sample size was 3016, were collected from CBMCBs outdoor and inpatient from ward during last 3 years. Picture showing some traditional culture equipment. How do we use that? This is also some traditional culture equipments. We collect sheep blood, sterile test tube and antibiotic panels. Automated culture equipment, that is BACTEC, we do not use this. Types of specimen was urine, stool, bone swab, sputum, CSF, pleural fluid, ascitic fluid, prostatic smear, high vaginal swab, throat swab. Sample are collected in the patient, outdoor patient of the CBMCB and indoor, where special culture media was not used. Antibiotic panels. We used 19 antibiotic, that is amikacin, amoxicillin, azithromycin, ciprofloxacin, cephradine, cefixime, cefuroxime, ceftriaxone, cotrimoxazole, cefalexin, colistin, doxycycline, gentamicin, imipenem, levofloxacin, meropenem, nalidixic acid, nitrofurantoin, and vancomycin. Media used for this isolation of bacteria. We used nutrient agar medium, McConkes agar medium, blood agar medium, chocolate agar medium, molar Hinton agar medium, and high chrome UTI agar medium. High chrome UTI agar medium is a chromogenic differential medium recommended for presumptive identification of microorganism, mainly causing urinary tract infection. Various kind of bacteria produce variety of color, and bacteria can be identified easily by comparing the color from chart. Picture showing this color chart. We used Dick's diffusion method for antibiogram. This method is based on inhibition of growth of test microorganism by using test compound in agar medium plate. Now, culture sample size. In 2021, sample size was 838. In 2022, it was 956, and finally 2023 sample size was 1222. Male and female culture study. Male was 693, that is 23% of sample size, and female was 2323, that is 77%. Now result, sample size was 3016, where culture positive was 641, that is 21.25%, and culture negative was 2375, that is 78.75%. Culture positive male and female. Out of 641 positive study, where male culture positive was 225, that is 35%, and female culture positive was 416, that is 65%. Now, gram positive and gram negative bacteria findings from culture positive study. Our gram positive findings, bacterial findings was 357 sample, which is 55.7%, and gram negative bacteria findings is 284, that is 44.3%. Isolated bacteria was, in case of gram positive, Staphylococcus species, Streptococcus species, Enterococcus species, which contain 55.7%. And in case of gram negative, it was Escherichia coli, Klebsiella species, and others, which contain 44.3%. Isolated bacteria. Left sided shows the gram negative bacteria, that is 44.3%, and right sided showing gram positive bacteria, that is 55%. Individual sample size. Urine sample, it was 2526, that is 83.75%. Stool sample was 55 in number. Own sample 247. Prostatic smear and hydrogenal swab was 63 in number. Pleural fluid, ascitic fluid, it was 31. Sputum sample was 22, and throat swab was 02. These are the 19 antibiotic panels we used. Now, antibiotic sensitivity and resistance pattern individually. First one is amikacin. Resistance was 18% and sensitive was 82%. Amoxicillin, resistance was 82%. Azithromycin, resistance was 84%. Ciprofloxacin, it was 56% resistance. Cefradin, 73% resistance. Cefixime, 78% resistance. Cefuroxime, 58% resistance. Ceftriaxone, 61% resistance. Cotrimoxazole, 70% resistance. Cephalexin 75% resistance, colistin 75.5% resistance, doxycycline 41.5% resistance, gentamicin 35% resistance, imipenem 2% resistance. That is the only antibiotic which is in good condition. Levofloxacin 50% resistance, meropenem 18% resistance, nalidixic acid 84% resistance, nitrofurantoin 16% resistance, vancomycin 79% resistance. Overall table showing overall antibiotic susceptibility and resistance. Table showing national antimicrobial resistance surveillance in Bangladesh. That was in 2016 to 2023 by IEDCR, showing this. Sample size was 24,837 from urine. Amikacin resistance 8%, penicillin 48% resistance, azithromycin 74% resistance, netilmycin 15% resistance, cefuperazone 20% resistance, cefixime 69% resistance, cefuroxime 66% resistance, ceftriaxone 53% resistance, erythromycin 89% resistance, moxifloxacin 27% resistance, polymyxin 27% resistance, cefepime 34% resistance, ceftazidime 36% resistance, imipenem 9%, sulfamethoxazole 48% resistance, meropenem 11%, nalidixic acid 72, nitrofurantoin 28, mesalinam 47%, and ofloxacin 41% resistance. Now discussion. The link between inappropriate use of antimicrobials and development of antimicrobial resistance was acknowledged in different scientific studies and global proceedings. In Bangladesh, prescribers generally diagnosed microbial infection on clinical judgment and select antimicrobials on empirical basis, which unfavorably affect the sensitivity pattern of microbes. Antimicrobial sensitivity pattern reflects the emergence of resistance against common antimicrobials, which are similar with that of other previous studies. Though antimicrobials like ciprofloxacin, ceftazidime, and ceftriaxone have become ineffective because of emergence of resistance, the microbes are still sensitive to imipenem, meropenem, nitrofurantoin, and amikacin by this study. Study from IEDCR, National AMR Surveillance Report Bangladesh, showed organisms isolated from urine and surgical wound infection were more resistant to ampicillin 93%, ceftriaxone 80%, and cefuroxime 90%. Alarming drug resistance of this study were as follows: amoxicillin 82%, cefradin 73, cefixime 78, ceftriaxone 61, cotrimoxazole 70, nalidixic 84, vancomycin 79, azithromycin 84, ciprofloxacin 56, cefuroxime 58, cephalexin 75, colistin 75, and levofloxacin 50%. Contributory factor may drug overuse, misuse, and inappropriate prescribing practices by physicians, along with intrinsic microbial plasmid mediated factors. An etiological impact of use of antibiotics in veterinary medicine as therapy or food additives may also cause drug resistance. This resistance bacteria and gene can be transferred from animals to human through the food chain. Moreover, increased population and urbanization can lead to increased dissemination of antibiotic resistance bacteria and resistance gene, which was also seen in other countries. It was a striking finding in the present study that ceftriaxone, cefuroxime, cefixime, ciprofloxacin, meropenem resistance rate increases in recent year. Now conclusion. Antimicrobial resistance is a complex global public health challenge, and no single or simple strategy will suffice to fulfill contain the emergence and spread of infectious organisms that become resistant to the available antimicrobial drug. So, right choice of antibiotic, maintenance of proper dose, duration, rational use of antibiotics in human and veterinary sector may improve this devastating condition. Finally, culture negative does not indicate there is no bacteria. Antibiotics a precious resource and handled with care. We need to preserve this resource by working together. Acknowledgement and references. We are thankful to doctors of CBMCB, faculty member of Microbiology Department CBMCB, and admitted outdoor and indoor patient of CBMCB. Thank you.